Assalamualaikum.
Waalaikumsalam.
Sing diombe kopi niku sapa? Wong silakan ngopi.
Jarene ngopi Diki. Ya sudah, tak silakan ngopi. Yang suka ngopi gitu loh.
Itu kok ada orang yang sama ya, namanya sama itu.
Iya ini loh.
Beda orang ya, satunya Muhammad Yunus, satunya juga Muhammad Yunus, ada dua itu.
Nanti tak keluar. Tenang saja.
Saya takut ada trouble, saya gunakan dua komputer.
Ayo ya kan? Ini live YouTube loh ya. Jangan guyon-guyon loh ya.
Oh iya, enggak ya, guyon-guyon enggak ngapa-ngapain.
Oh iya ya, bener bagus.
Cuman yang ini saya mungkin sih ini ada backgroundnya ini. Oh iya, aslinya. Yang satunya sih coba saya perbaiki.
Assalamualaikum.
Waalaikumsalam.
Lagunya belum ya.
Hari
Ibu itu baca apa itu Bu Endang? Katanya kok serius banget dari tadi baca film gitu ya.
Prof Makoto sudah hadir, Prof Makoto.
Oh gitu ya?
Itu kelihatan. Ketemu informasinya.
Assalamualaikum.
Happy birthday. Sehat selamanya.
Oke, sugeng milad, Dokter Rima.
Terima kasih, Yunus. Halo,
selalu ya. Sukses selalu.
Thank you very much.
Bu Rima.
Good morning. Terima kasih.
Makoto.
Ohayou gozaimasu. Oh, genki desu ka?
Genki desu.
Hai.
Ayo.
I'm fine.
Fine. Okay.
Oh. Selamat pagi, Dokter April.
Oh, Dokter April juga sudah muncul.
Assalamualaikum, Dokter April. Sensei, nihon ni wa mada samui desu ka ne?
Samui desu.
Sou desu ka ne?
About 60 degree.
Oh.
Very cold.
Pada samui ne.
Saja gitu artinya itu.
Selamat pagi, Dokter April. Selamat pagi, Prof Lucy.
Assalamualaikum, Dokter April.
Waalaikumsalam, Prof Yunus.
Dokter April,
apa kabar, Dok? Alhamdulillah sehat. Prof Makoto, Matsubayashi, how are you? I'm fine. I'm so glad to see you here. Yes. You attended to this lecture. Yeah. How are you? About your family all? That's good. Everything is okay. Okay. How about Hinoka and Yotaro? Everyone is fine. Okay. How about the infection of the COVID-19 in Indonesia? We are heading to endemic, I think. I hope. Have you all vaccine already at 3 times?
Already.
Yeah.
Until three booster.
Oh.
Yeah? 3 booster. Yes.
Very good.
Very good.
But side effects are so bad. How about side effect?
Selamat
Side effect.
pagi,
Yes.
good morning.
Pagi, good morning.
Morning.
Mas Bro.
Good morning.
Assalamualaikum, Mas Bro.
Thank you very much.
Dokter April, ada amunisi baru ya sekarang masuk. Amunisi baru dia itu ya.
Ya,
Masih mute, Dokter April.
Betul. Apa namanya? Kita sebenarnya happy ya ada dia bisa masuk ke Balit Vet. Tetapi masalahnya sekarang kan kita itu sudah pindah ke BRIN.
Oh iya.
Ke BRIN. Jadi jadi untuk CPNS yang baru masuk itu tidak bisa menjadi peneliti.
Masuk.
Oh, sayang ya.
Sayang sekali sih, benar
ya,
sayang.
Dia
gitu
backgroundnya peneliti.
Betul. Dia kan pengen sekali jadi peneliti kan.
Iya. Terus jadi apa? Administrasi aja.
Ya mungkin bisa jadi penyuluh, bisa jadi analisis kebijakan, bisa jadi
lebih ke arah itu ya.
Paramedik yang model-model seperti itu. Jadi tidak bisa menjadi fungsional sebagai peneliti.
Fungsional peneliti.
Betul, karena BRIN itu sudah menutup per Januari 2022 itu nama terakhir yang bisa masuk ke BRIN gitu. Ya, sementara kan teman-teman CPNS ini baru masuk kemarin Maret, awal Maret ya, Februari lah, akhir Februari gitu. Jadi tidak bisa. Kalaupun bisa, mungkin nanti dalam bentuk dalam sistem mutasi gitu.
Oh, waktu
Kalaupun
pengumuman penerimaan rekrutmen itu memang disebutkan ya seperti itu ya.
kalau waktu
sampai
rekrutmen
ke
memang masih valid vet, Prof.
masih valid vet.
Kita itu pindah
Iya,
ke BRIN per
iya.
1 Maret kemarin.
Jadi valid vet sudah tidak ada ya?
Sudah tidak
Gimana
ada.
gitu? Tidak
Sudah
ada.
tidak ada.
Istilahnya BRIN ya, langsung BRIN ya.
Langsung BRIN, iya.
Supaya ini terlibat
Jadi
dalam riset ya.
namanya Pusat Riset Veteriner sekarang.
Pusat Riset Veteriner.
J.
Sehingga tapi bisa join ini nanti bergabung.
Oh, bisa.
Kolaborasi riset
betul.
ya.
Lebih open sekarang.
Lebih open. Ya.
Untuk riset.
Oh, gitu ya.
Sayang sih sebenarnya.
Sayang ya. Dia memang belajar banyak di waktu itu S2-nya sampai dia belum mendapatkan waktu PNS yang tahun kemarin kan ditolak ya, belum diterima, sehingga dia sudah experience-nya udah bagus ya.
Iya.
Tapi enggak apa-apa lah. Masih bersyukur lah. Bisa PNS itu kan dicari.
Orang susah sekarang,
Susah. Prof Makoto, how about quarantine and COVID quarantine in Japan?
Quarantine?
Quarantine in Japan.
Quarantine.
Yes.
If there is some foreigner
Ah,
go to Japan.
Yes.
How long?
Now, today, maybe 1 week.
1 week. I see.
Yes, short term.
In the hotel, yeah?
Yes.
So.
I don't know, good hotel or not.
Still like that, Prof.
Precious and delicious.
Yeah.
So your institute, your university has open for student at the moment.
Yes. But some students were infected with COVID-19. Then we must close only the laboratory or room and so on. And we must sterilize using the ethanol.
Ethanol. And Omicron. How about Omicron? Yes, same.
Yes. The most virus genotype is Omicron in Japan also.
Okay. Yeah, we wish COVID-19 decrease, and I can to go maybe to go to Japan. Terima Dr. Lucia, Prof. Lucia.
Now it is very difficult to go abroad from Japan to other countries.
That's the Japanese embassy is now already open to get to release some visa from the foreigner, because some our young teaching staff will go there, so that already have the visa from Japanese embassy. Yeah, next week I think she will go to Yamaguchi or somewhere. Even not so easy to get some visa, because it's very strict to the requirement to some. I don't know how many times vaccination to requirement to go to Japan. 2 times or 3 times basically.
Maybe yes, 2 times or 3.
I see.
And the certification of the negative by.
Yeah. Good.
Pak Yunus sudah diingatkan sama Bu Puji, sudah jam 9.
Okay.
Silakan Dr. Adit.
Ya. Assalamualaikum warahmatullahi wabarakatuh. Good morning and
Good
warm greeting
morning.
to everyone. Hope you are fine and safe during the pandemic situation. First of all, let us pray to Allah Subhanahu wa Ta'ala. Because of His bless and mercy, we can meet together without any obstacle with healthy condition. The Honorable Professor Dr. Mirni Lamid, Doctor of Veterinary Medicine, Master of Agriculture, as Dean of Faculty of Veterinary Medicine, Universitas Airlangga. The Honorable Dr. Rimayanti, Doctor of Veterinary Medicine, Master of Health, as First Vice Dean of Faculty of Veterinary Medicine, Universitas Airlangga. The Honorable Professor Makoto Matsubayashi, Doctor of Veterinary Medicine, Doctor of Philosophy, as a keynote speaker from Osaka Prefecture University, Japan, also as our respected colleague who already contributed previously in Adjunct Professor program in 2019 until now. Also, encourage collaboration in publishing many scientific articles in various reputable international journals. The Honorable Lecturer from Faculty of Veterinary Medicine, Universitas Airlangga, the Honorable Student from Osaka Prefecture University, Japan, and also all of students from Faculty of Veterinary Medicine, Universitas Airlangga. Ladies and gentlemen, firstly, let me introduce myself. My name is Aditya Yudhana as Master of Ceremony, and welcome to our event, Guest Lecture Adjunct Professor, which organized by the Division of Veterinary Parasitologi, Faculty of Veterinary Medicine, Universitas Airlangga. For your information, today's event also available on YouTube live streaming and already attended with more than 270 registered participants, which consisting of Indonesian Veterinary Parasitologi Association or APARVI, Indonesian Society for the Eradication of Parasitic Disease or P4I, undergraduate student, postgraduate student, veterinary co-assistancy student, lecturer, researcher, practitioner, and other colleagues, not only from Faculty of Veterinary Medicine, Universitas Airlangga, but also from many other universities. Before we come to the main session, let's start by praying first, so the event on this day will run well without any obstacle at all. Pray based on individual belief. Pray begins. Finish? For the first agenda, let's sing the national anthem Indonesia Raya together and followed with Hymne Airlangga. For all participants, please stand by in a straight position. And for the operator, the time is yours.
Come on.
Okay, thank you very much. We're sorry due to technical sound problems. Okay, ladies and gentlemen, on this great occasion, let me deliver the structure of the event today as follows. The first one, opening remark. The second agenda, speech from Dr. Rimayanti, Doctor of Veterinary Medicine, Master of Health. The third agenda, general lecture, which will be delivered by Professor Makoto Matsubayashi. The fourth agenda, question and answer session. And the last one is closing. Now we would like to invite Dr. Rimayanti, Doctor of Veterinary Medicine, Master of Health, for the opening speech. To Dr. Rimayanti, Doctor of Veterinary Medicine, Master of Health, the time is yours.
Yeah, thank you very much, Dr. Aditya Yudhana. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam.
Waalaikumsalam warahmatullahi.
Good morning, ladies and gentlemen. Before we start the program, let us praise and thanks to Allah Subhanahu wa Ta'ala, who has been giving us blessing and mercies, so we can attend a guest lecture program this morning in good condition. I would like to welcome Honorable the Head of Veterinary Parasitologi Division, Faculty of Veterinary Medicine, Universitas Erlangga, Dr. Pujiastuti. And then on behalf of the Faculty of Veterinary Medicine, Universitas Erlangga, I would like to welcome our honorable guest lecturer today, Professor Makoto Matsubayashi, DVM, Ph.D., lecturer from Osaka Prefecture, University, Japan. We welcome Prof. Makoto Matsubayashi to the virtual classroom of Faculty of Veterinary Medicine, Universitas Erlangga, Surabaya, Indonesia. Ohayo gozaimasu, Professor Makoto. Oh, genki desu. Yeah, we are very glad to meet you this morning. I hope Professor Makoto in good health, stay safe and keep healthy always, Prof. Makoto. And then I would like to welcome our honorable professors, lecturers, and students of Faculty of Veterinary Medicine, Universitas Erlangga, and all participants who might join the guest lecture this morning. It is my great pleasure to welcome all participants who have joined this guest lecture today in our virtual classroom. Thank you very much for joining this event. Ladies and gentlemen, today we attend guest lecture speech about molecular identifications of gastrointestinal protozoan parasites in livestock and successful trials toward one-cell analysis. Even though these lectures and transfer knowledge are still given online, hopefully all of us can obtain a great knowledge. So we hope that we can learn a lot from the lecture session, and we will have interesting and stimulating discussions. I encourage all of the students to ask questions and discuss about this interesting topic. Ladies and gentlemen, we know that the pandemic has been running for 2 years. March 17, 2022, exactly 2 years since Universitas Erlangga has restricted activities at campus. But of course, there is a blessing in this case, in this pandemic situation, because we can make the online teaching more interesting by featuring guest lectures from other universities around the world, and of course, make it easier to us to attend this meeting by online system. But much more than this, we hope the pandemic will be over, will end soon, and we can meet face-to-face again, offline, and return to the normal situation. Amen. Ladies and gentlemen, in this very good occasion, I would like to thank the head of Veterinary Parasitologi Division, Faculty of Veterinary Medicine, Universitas Erlangga, Dr. Pujiastuti, and all of the lecturers in the division for organizing this event. And of course, very warmly thanks to our speaker today, Professor Makoto Matsubayashi, for delivering such important and interesting topic, so all of us can improve our knowledge. Domo arigato gozaimasu, Professor. But I sincerely hope all the participants will enjoy the lecture, and I wish the program will be fruitful for us. Thank you very much. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam.
Thank you, Dr. Aditya.
Yep, thank you very much. Thus, the remarkable speech delivered by the first Vice Dean of Faculty of Veterinary Medicine, Universitas Erlangga. Thank you very much to Dr. Rimayanti, Doctor of Veterinary Medicine, Master of Health, for the opening speech.
You're welcome.
And now, before we move into the next agenda, please allow me to take a picture from all participants. So kindly reminds to all participants, now you can turn on your camera or video picture. Okay, we will take screenshot and starting countdown from 1 to 10. 1, 2, 3, 4, 5, 6, 7, 8, 9, and last one, 10. Okay, thank you very much. On behalf of committee, now we proudly present a short video profile which will be guided by Professor Dr. Lucia Triswanti, Doctor of Veterinary Medicine, regarding the collaboration which already settled between Division of Veterinary Parasitologi, Faculty of Veterinary Medicine, Universitas Airlangga, with our respected colleague, Professor Makoto Matsubayashi, Doctor of Veterinary Medicine, Ph.D. from Osaka Prefecture, University, Japan. For the video profile operator, the time is yours.
Thank you, Dr. Adit. Professor Makoto Matsubayashi as our collaborator since 2019. Thank you, Dr. April, for introducing us to him. And this is the activity Prof. Makoto at first Adjunct Professor. He gave a guest lecture and also training, and it's with participant after the guest lecture. Next, and this photo discussion with veterinary parasitology staff and division parasitology. It's also Prof. Makoto Matsubayashi discussed with our magister student. It was planned that they would be recruited to participate in the Sakura project, but unfortunately it didn't happen because the pandemic. It's also after give a lecture and training. This is a photo session after training in laboratory parasitology with magister student and also PPDH student. It's Prof Makoto and Prof Yunus and me. It's a training diagnostic of the gastric tract protozoa, especially in Cryptosporidium and Giardia, with IPKMV Magister student. It's Prof Makoto explain how to account OPG with Dr. Endang Supriyati. This is Prof Makoto when gave a training. It's IPKMV Magister student practice with his training. Also with Dr. April. Dr. April always join us when Prof Makoto in Surabaya. Thank you, Dr. April. This photo take at molecular laboratory with gas from, I forgot, it's from Nigeria or another place in Africa. Is Prof Makoto explained the method to our student in laboratory biology molecular. With veterinary parasitology staff, Prof Makoto and Dr. April go to Bangkalan Livestock Service and discuss with staff in Bangkalan Livestock Service and also see Madura cattle. After this, it's a photo. Prof Makoto see Madura cattle. I think that's all activity in 2019. Because pandemic, Adjunct Professor was not held on 2020, but postponed until in 2021. This is Professor Dr. Mustafa Helmi Effendi open this event. And this event was moderated by Professor Yunus. This is some participant when we had Adjunct Professor in 2021. And with his trademark smile, Professor Makoto start to sharing knowledge with the topic gastrointestinal protozoan parasite as important factor for reducing productivity in livestock. And on his PPT, he also include our photo when we visit to Opu. Thank you, Prof Makoto. And There are some protozoan in livestock. This is Dr. April join with us, and he is Prof. Makoto best friend. So if Prof. Makoto in Indonesia, Dr. April always join. And Adjunct Professor at 2021, closed by Prof. Nunu with sending certificate to Professor Makoto Matsubayashi. I think that's all. Let's we follow the guest lecture now. Thank you, Dr. Adit.
Thank you very much, Professor Lucia. And now, before we get started, I would like to inform several points so you know how to participate in today's lecture, which held online via Zoom. You have joined the presentation by using your own phone, tablet, or computer. All participants may not activate their microphone feature until we allow you to do so. In your Zoom control panel below the screen, you can find chat box feature. So for the general question related with this lecture main topic, please write down your question in the chat box. Moreover, you may submit your question at any time during the presentation. Then we will collect and address them at the end of the presentation. In this session, we have a remarkable international lecturer. He is Professor Makoto Matsubayashi, Doctor of Veterinary Medicine, Doctor of Philosophy from Osaka Prefecture University. Professor Makoto Matsubayashi will deliver insightful topic in today's lecture entitled Molecular Identification of Gastrointestinal Protozoan Parasites in Livestock and Successful Trial toward one-cell analysis. Before we begin the lecture session, I would like to invite our remarkable moderator. He is Professor Muchammad Yunus, Doctor of Veterinary Medicine, Master of Health, Ph.D. Professor Muchammad Yunus was born on 29 December 1966 and got his Doctor of Veterinary Medicine degree in 1992, then got Master of Health degree in 2001. Also, Professor Yunus has got overseas scholarship to continue his doctoral degree and graduate as Ph.D. from Yamaguchi University, Japan. Until now, Professor Yunus working as a professional lecturer and professor in the Division of Veterinary Parasitologi, Faculty of Veterinary Medicine, Universitas Erlangga, with more than 30 scientific publications and professional experiences in both national and international. Without any further ado, ladies and gentlemen, please welcome our moderator, Professor Muchammad Yunus. The time is yours.
Okay, thank you, Mr. MC, Dr. Adit. Okay, Assalamualaikum warahmatullahi wabarakatuh. Good morning, everybody. So, ladies and gentlemen, today I will try to be here to deliver this event. And this morning, I hope everything is going well with you all, the participant, and also the honorable our guest lecturer, Professor Makoto Matsubayashi. Good morning. Ohayou gozaimasu, sensei. Okay, today guest lecture or webinar. So the topic is very important for us because this is the good opportunity, good chance for all participants from undergraduate student, master, doctoral program, and also lecturer, practitioner, and every people who interested in this topic. because we hope everything by sharing knowledge from Professor Makoto Matsubayashi is very important to improve our knowledge. Okay, ladies and gentlemen, before Professor Makoto Matsubayashi deliver his topic, just I will read to some the curriculum vitae, his curriculum vitae. Professor Makoto Matsubayashi, he was born in Japan, Osaka particularly, in October 23, 1973. So still young, yeah? And then his background education is he's graduate from undergraduate from Department of Veterinary Medical Sciences, Faculty of Agriculture, Osaka Prefecture University, 1999, and he's graduate from Osaka, also the same university, in 2009, Doctor of Philosophy, and then so many experiences, work, and also research and publication from 1999 until 2000, until now, I think. And the peak career is in 2020. He is got from professor, is become professor in Department of Veterinary Immunology, Faculty of Environmental Sciences, Graduate School of Life and Environmental Sciences, Osaka Prefecture University. So many experiences in research and publication. If I read publication, around more than 150 publication, international and national publication, and reputable journal. And ladies and gentlemen, I think now we will hear and learn about the topic from Professor Makoto Matsubayashi. And then now I would like to invite Professor Makoto Matsubayashi, DV, Ph.D, deliver of his topic, your topic, and please welcome Professor Makoto Matsubayashi. And I would like to remind you about we have limited time, around maybe 1.5 hours, so that if there is some question from all participants, will submit in the chatting box. Okay, please welcome Professor Makoto Matsubayashi. The time is yours.
Thank you very much for kind and very detailed introduction of me. First of all, I'm expressing my great acknowledgement to all of professors and staffs. Actually, I'm looking forward to having a lecture today and seeing you all. So I would like to talk about the gastrointestinal protozoan parasite using my slide. I will show you. Okay, so I will start my lecture. Actually, I'm very sorry. I couldn't go to Surabaya, Indonesia, due to the COVID-19. Maybe it's 2 years. I couldn't go there. So I'm very missing, and but I can see you again today, even in Zoom. I'm very happy. So I will talk about molecular identification of gastrointestinal protozoan parasites in livestock and successful successful trial toward one cell analysis. Okay, shall we start? Wait. Okay. Actually, already Dr. Yunus or Lucia introduced me, so I will shortly introduce myself. I was born 1973 years. Yes, I'm still young. So I was born Osaka, Japan. So my family is only one wife, actually, officially, and one son and one daughter. One son is about 11 years old. One daughter is maybe 4 years. And my hobby is fishing, like that. I go sometimes to harbor, and I'm doing fishing, and movie viewing, and driving, and yes, microscopic observation, and finally visiting Indonesia.
Ah, good.
Yes. Now I'm often watching Indonesia in YouTube. I and my son and daughter singing a song. Indonesia anta mahaluku. Yes. Now I just watching Indonesia in YouTube. So about educational background, Dr. Yunus already introduced. Then I will omit. This is the picture when I was undergraduate student and when I was a younger researcher. So this is the map of Japan. I'm here. This is Osaka. Actually, Osaka is located here. Tokyo is here. So Osaka is the second largest city in Japan. Of course, the first one is Tokyo. Then Osaka Prefecture University is located here. Actually, this is Kansai International Airport. Kansai International Airport is located around here, south area. So Osaka Prefecture University is here. So that is why if you or student come to Osaka Prefecture University, you can easily move from airport to university. About 3 or 4 minutes by train. Only one station. These are my students. Some students are foreign students from other many foreign countries. So this is my profile. I will introduce shortly. After graduation, I entered to Osaka Yuhigaoka Gakuen Junior College. I worked as a teacher, assistant professor, lecturer, and associate professor, and so on. Then I moved to institute. The name of the institute is Department of Parasitologi, National Institute of Animal Health, National Agriculture and Food Research Organization. This is a government institute. So maybe the similar with the Institute of Dr. April.
Nice.
Yeah. Then during working at the institute, I stayed in Subang in Indonesia as a JICA parasitology expert. This was the first visiting to Indonesia. I stayed at Surabaya for several months. This is Kota Bandung. Then after that, after institute 2016, I moved to Osaka Prefecture University as a teacher. So then 2 years ago, I moved to the laboratory of veterinary immunology. But my research, special research, is about parasitology. Then, from 2017 to now, I'm doing project about protozoan disease with Dr. April from Bogor, and collaboration with Universitat Airlangga, Dr. Nunuk and Lucia, and other professors. Finally, I have been to Indonesia 16 times.
A lot of experiences.
And now, for example, this is a picture for staining at Sudan. This is me, and this is Dr. Sylvia. She was already, also graduated from Airlangga University. At that time, she was the counterpart of me. We were worked at the field for Bradley protozoan parasite or gastrointestinal parasite, like that. So we often went to the survey for targeting intestinal parasite. Yes, this is also a picture. My sincere feeling to Indonesia. I'm loving warm and kind personalities of Indonesia, the best. So nice food, of course, food and hard-working students, and so on. That's why I'm always getting fat, even during my short stay in Indonesia. Actually, additionally, I have many friends. I have 3 Agus and 4 deer as my friend. If I receive the email from deer, but I don't know who's deer. That. Yes, and this is also professor's pictures of Airlangga University. At that time, I was so happy and I was so pleased because many professors came to my university. But I'm waiting for the next visiting. Okay, then today's top. This is today's top agenda. So first, I will talk about shortly general information of intestinal parasites, and second, selected important protozoan parasites. I would like to focus on 2 parasites. One is Cystoisospora suis in pigs, then Endomember species in pigs. Each topics or each protozoan parasite, I will talk about general information, how to detect, significance of genetic analysis and protocols. I will show you the detailed protocols. Then finally, one cell molecular analysis for parasitology. Maybe after today, you can be the specialist of protozoan parasites. Hey, from now, I will talk about general information shortly. Swine and cattle and chickens. Maybe most of the students know very well about these parasites. Do you know all? For example, stomach, kidney, ticks as ectoparasites. In meat, we can find Taenia solium or Trichina species. In lemon, Trichuris suis, Oesophathomonas, and so on. Lung and liver, we can find Ascaris suum, and so on. And in small intestine, these parasites can infect pigs. For example, Ascaris suum, Strongyloides, and Eimeria, Cystospora, Cryptosporidium, and Endomember, and so on. Please remember red color. Please remember some parasites as red color here. About bovine parasite, of course, ticks and eyeworm, Taenia saginata, fasciora, and so on. We can find Dictocalus in lungs and these parasites. Then, as important protozoan parasites, which I show red color. But we can find some parasites in small intestine, Cooperia and nematogears and bonostomum and so on. But we want to focus on Giardia, Hymeria, Cryptosporidium. These are sometimes important for livestock production. Then in poultry parasites, Signabass and Capillaria, and inbred parasites, we can find Leucostozoan, Plasmodium, Hemoproteus. Of course, these broadly protozoan parasites are important in chickens and liver, Histomonas meliagridis. In small intestines, we can find Hymeria, Ascarididae, Capillaria, F1, and so on. Maybe students know very well about all parasites. Then, what is the common point? I marked red color in the list. So, among the red color protozoan, they are very small and difficult to be detected in feces or from feces by microscopic examination. And they are just simple shape and difficult to be differentiated from other debris. Sometimes fungus or pollen and so on. It is sometimes difficult to differentiate these are debris or protozoan parasites. And additionally, they are almost impossible to morphologically identify the species or genotypes. For example, they are Hymeria tsuerni or other species and so on. It is very difficult to identify exactly. We can say it seems to be Hymeria tsuerni, but it is we must check in detail to exactly identify the species. So I often found some authors actually identify the protozoa under microscope by mistake. I review some papers in 1 month, but in some papers, authors made mistake the identification of the parasite. For example, the student said to me, "This is Cryptosporidium oocyst." I will ask, "What evidence?" This is round and very small. You can just say this may be Cryptosporidium because we must check and confirm by several methods. So what are we doing in such a case? We must know well first how the parasites are morphologically seen on the slide under microscope. For example, we must know what the color and oocyst or cyst wall, thin or thick or very rough or smooth, and contents. We can see the residuum or sporocyst or sporozoid or one cell or proliferated cells and so on. But not only shape. We must know in detail information. And then how the parasites are confirmed, we must know. For example, to confirm the species or parasite, we must do sometimes specific staining or immunostaining or molecular analysis and so on. Only morphological data are sometimes risky to be correctly identified and can lead misunderstanding of the infection in the host. So I will talk about the impact of the gastrointestinal parasite especially protozoan parasites. GI, nematodes, trematodes, and cetodes. Of course, protozoan parasites are only nematodes, trematodes, and cetodes are generally comparatively thought to be less pathogenic and can induce economic losses in livestock production, as you know, by significant decreasing weight gain and milk production and in chickens, egg productions. However, protozoans tend to cause, compared to these gastrointestinal parasites, protozoans tend to cause clinical symptoms such as diarrhea, resulting in sometimes bloody or lethal or severe cases as a major negative impact. They can sometimes related to the index for poor farm management because these oocysts or cysts or eggs can be transmitted by fecal oral route. That is why if we find some parasites, that means fecal contamination is occurred there, indicating fecal contamination because most of them are resistant, strongly resistant to environment and disinfectants. That is why in the farm, if some host infected with these parasites, it is very difficult to clean up the parasite in the field or farm. Recently, some of protozoan can also infect human as zoonotic impact and have a potential impact as zoonotic. So recently, the Ministry of Agriculture in Indonesia launched a revitalization program to reduce food imports and increase domestic productions based on policies of the government. What can you do as veterinarian? For example, maybe just I read in the book, Indonesian government wanted to reduce the import from other countries. So they suggest the food or many things are produced in their own country. So, but otherwise, at each farm, for example, the same farm and farmers are use the same food like this. But if they don't care any infection of parasites or don't care hygiene programs, maybe the same food, but income will be different. For example, the farmer can obtain huge money, but they are use the same food, but they can reduce the income of the money like that, because cost of treatment and prevention to disease such as diarrhea can reduce, and then lower weight gain, productivities of egg and milk can be occurred. So we move to the details information. I focus on selected important protozoan parasites. First, Cysticercospora suis in pig and Endomember suis in pigs. Here, I just introduce only 2 species, but after my talking, student or student can apply this methodology or logic to other protozoan parasite or parasite even in virus or bacteria and so on. So, swine industry in Japan. This is just the introduction of the situation in Japan. In Japan, so produces 10 to 14 piglets, which mean at 18-25 days. They are shipped in 6 months in Japan to slaughterhouse. Then they are almost 100 kilograms. So we must care piglets, especially from various infectious agents, because 6 months is very short. That's why if some infectious agent affect, maybe the weight gain can reduce dramatically. Infectious agents, including protozoan parasites, which cause diarrhea. Actually, they are born in the same day, but some piglets are very thin and small, very low weight gain, like that. We call round piglets. So, of course, the pathogenic agents causing diarrhea are not only protozoa. Of course, as a veterinarian, we must know all of the pathogens causing diarrhea. For example, bacteria, Escherichia coli, sometimes toxin-genic intestinal agglutinations, adhesive genes, and so on. Salmonella and Lawsonia intestinalis, intracellularis. This bacteria cause proliferative enteritis in pig, and so on. And Brachyspira, and so on. Then virus, as you know, swine enteric alpha coronavirus, rotavirus, and so on. But in my opinion, these bacteria and virus can be detected easily because the protocol are fixed, and then we just do according to the protocol. That's why maybe it is relatively or comparatively easy to be detected. But in case of protozoan parasite, for example, Cystoisospora suis and Endomember, and then we want to add it, Cryptosporidium. It is sometimes difficult to identify or detect correctly. That is why we must diagnose carefully. First, I will show you Cystoisospora suis. As you know, this parasite can infect piglet or pigs. This is a life cycle. Oocyst of Cystoisospora suis infected by oral route. Then the parasite, especially sporozoite, can be released from the oocyst and sporocyst. Then sporozoite can invade into intestinal radiculopia. So the sporozoite can proliferate as asexual development, as melons or seasons. And then they can develop sexual development. For example, microgametocyte or macrogametocyte. And then for fertilization, and then many oocysts are produced in feces. Of course, during the development, the host can show diarrhea or severe or lethal diarrhea. Then in feces, we can see the oocyst. But as you know, unfortunately, pigs can be infected with Cystoisospora and Eimeria. Usually, cattle can be infected only Eimeria. Chicken also infected only Eimeria. And maybe Calvinola. For example, dog or cat can be infected with Isospora or Cystoisospora. But pigs are very interesting. Pig can be infected with both Eimeria and Cystospora. So you can see Cystospora contain 2 sporocysts and 4 sporozoites in each sporocyst. But Eimeria, the oocyst contain 4 sporocysts and 2 sporozoids. Total 8 sporozoids are the same, but different, the number of sporocysts and sporozoids. But we find the oocyst in feces only one cell. That's why we can differentiate which is the oocyst, Cystospora, and Eimeria. That's why we can differentiate the species only after sporulation. So we must count the number of sporocysts or spores. But it's very tough, and it takes much time to sporulation, for sporulation. That's why we can do anything. Actually, cystosporocysts can cause lethal or diarrhea, but 7 Eimeria species in pig are thought to be less or no pathogenic. That is why it is very important to differentiate or detect or identify the species of Cystospora. I will show you some cases. For example, in case 1, in 2014, 6 piglets were dead suddenly in farm of Japan. So veterinarian of local government did necropsy and pick up some organs, and they made section and stained by hematoxin eogen, HE staining. So veterinarian found the surface of intestine were disrupting, not normal like that. And then they successfully found some parasite in lamina propria, for example, like this. And by electron microscopy, they can find some zoites. How do you think this parasite? Actually, we can find some parasitophorus vacuole. We can see white area, and then zoites. This is the remarks of the infection of protozoan parasite, but we couldn't identify the species of these zoites. How do we analyze? Then we try to analyze genetically, but we don't have any feces, remaining feces. That's why we cut the section of the formalin fixed blocks here. Then I cut 5-10 sections at the thickness of 5 micrometers. And then we extract the DNA from the section. And we conducted the PCR. But we must select the good primer. So we searched which is the better primer by PubMed or other published journal. So we could find only one primer pair, ITSGF, ITSR2. If we use the primer, we can find the amplification by 450 base pair product. This product is contained partial portion of the ITS1 liposomal DNA locus of Cystospora species. But in this publication, they designed the primer for oocyst. That's why we tried to analyze to detect the Cystospora by section. Then, as a result, we can find clearly 450 base pair product here from the DNA of this section. Then we read the sequence of the product. So we construct the phylogenetic tree. So we can find our isolate here. This is the cluster of cystospora. These are peak. Then we can successfully and clearly identify the parasite as cystospora suis. So we don't say maybe these are cystospora suis, or I expected these are cystospora suis. No, we can confirm. We could confirm these are cystospora suis and causing lethal diarrhea because 6 piglets were dead. Yeah. And the second case is in 2016, 6-day-old piglet had gray watery diarrhea and emaciation and rough coat and labored breathing and difficulty standing. They couldn't stand easily, and they laying down. And some of piglets were dead. That's why the veterinarian of the local government did necropsy. So in intestine, they could find thinning of small intestine wall and some getting bigger. Then the veterinarian found the parasite here, like that, in the epithelium cells of intestine organs. And they found some merozoite and maybe trophozoite. And by electron microscopy, they found the zoetes. And additionally, you can see white area. Of course, white area and vacant area, they must be Parasitophorus vacuole. Then the government officer asked me how to detect or identify these parasites. Then I tried to do analyze again by molecular analysis. Formally fix block veterinarian cutting section of the thickness 5 micrometer, and they sent to me the tube of 5 sections. I extract the DNA from the tissue, and then I used the primer, the same primer. For example, ITS1 ribosomal DNA locus of Cystospora species for oocyst. He designed. But I want to analyze different gene locus by PCR. This is ITS1 gene. Then we searched sequence, published sequences. Then we could find the Cystospora sequence in gene bank. Then we newly designed cytochrome C oxidase subunit 1 genes. I originally designed the primer set of COX1. So we can successfully determine the sequence of C. suis in COX1 region firstly for the first time in Japan. That is why we can determine 2 genes in ITS1 and COX1 successfully. That's why I could send back the information that these parasites were Cystospora suis. So actually, brief summary. This is brief summary. This confirmation by PCR analysis or determination of the novel sequences, we can submit something new to the international journal. Maybe this is case 1 and this is case 2. The molecular identification of C. suis have been rarely done worldwide. Thus, I have reported the utility of the analysis in two papers of international journal like this. So maybe some students asked me, how about the Eimeria in pigs? Maybe 7 Eimeria species. We could successfully identify cystosporosis in pigs. How about Eimeria? Of course, I tried to analyze Eimeria species in pig when I was young, but very, very difficult. Previously, I tried to genetically identify Eimeria species in pigs, but it was so difficult, and finally I gave up. Maybe Eimeria species in pig were less significant. That's why I will try again in the future. But it is important to identify cystosporosis because the species are more pathogenic and virulent. Okay. Okay, that's all about cystosporosis. Then we move to the other parasite, Endomember in pigs. Okay, I summarize the Endomember species of the pig in this slide. In pig, 3 species of Endomember were well known. One is Eimeria, Endomember histolytica. As you know, these species are zoonotic, and in human, Endomember histolytica cause severe or lethal and very important symptom. So, Endomember histolytica can infect human many variety of many animal species, including pigs. And then parasitic site colon, of course, trophozoites can invade into liver and lung and brain. Very, very severe symptom. And then in pigs, Endomember polecki and Swiss. Totally 3 species can infect pig. But Endomember histolytica, of course, logically or experimentally, Endomember histolytica can infect pigs. But we have never found natural infection of Endomember histolytica in pig. That is why, in my opinion, Endomember histolytica can rarely or seldom infect pigs. Of course, pig possess sensitivity to the Endomember histolytica because experimental infection can be successful. So, how about the other 2 species, Endomember polecki and Swiss? In some papers, these 2 species are less or non-pathogenic. I could find this description in some published papers. So, Endomember polecki, the host of Endomember polecki are human and pigs and so on. That is why Endomember polecki are zoonotic. And Endomember Swiss infect only pigs. So parasitic sites are the same colon. And how about the morphological feature? Number of nuclei, this is 4, and these are 1. But in Endomember polecki, 2 or 4. And size are very similar. And trophozoites are very similar. That is why we can rarely identify by the data of morphological features. Then, do you know Stenbold, Dr. Stenbold? He is the first researcher to analyze genetically about Endomember. He reported the phylogenetic position of pig Endomember. So Endomember Swiss is here, and of course Endomember histolytica is around here, very far from the pig entamoeba. And entamoeba Swiss are here, and Chatoni, Porecki from human, and Porecki from pig. Entamoeba strontionis from ostrich. Each species are reported based on morphological data and hosts. But Dr. Stenbold suggests these are the same species. That's why Entamoeba porecki ST subtype 1, 2, 3, 4. That's why maybe pig can be detected as Entamoeba ST1 and so on. That's why we can see 3 species from pig, but Entamoeba histolytica is very rare, and Entamoeba porecki and Swiss. But porecki include 4 subtypes by molecular analysis. Then I could find the Entamoeba porecki and Swiss can be cause lethal pathogenic symptom. I will explain how to approach the pathogenicity of Amelia species and so the Entamoeba species. Actually, 1, 2, 3 prefectures, veterinarian of local government asked me how to detect. Actually, piglet at 3 prefectures show refractory diarrhea in 3 prefectures: Nagasaki, Aichi, Shizuoka. Of course, the veterinarian of local government analyze carefully by histological analysis and bacteriological analysis and virological analysis, then parasitological analysis. Of course, we must delete the possibility of these pathogenic agents. So this is the general information of Entamoeba species, because we must know how to diagnose or detect of the Entamoeba. For example, pig or piglet shedding are shedded feces. Then trophozoite or cyst. Entamoeba possess 2 types of the zoites. One is trophozoite and the other is cyst. What kind of trophozoite? What is the feature? Trophozoites are sometimes invade into intestinal mucosa, and trophozoites cannot survive in environment. That is why we cannot reserve the feces for diagnosis of trophozoite. That's why we must immediately make the fecal smear, and then we must check trophozoites as soon as possible. They can be detected by direct smear, of course, using fresh feces, but sometimes difficult to be diagnosed. As about the other type, the cyst can be infected stage, as you know. They can survive in environment for a long time and possess strong resistance to disinfectants. And cysts are detected by flotation method, relatively easy, but we had better stain by iod staining, like that. So how can we diagnose Entamoeba? Of course, using genomic DNA, we can do PCR analysis. For example, tissue, maybe trophozoite and cyst can be contained, and they are feces contain 2 parasites. That's why we can use or target the 2 types of cyst or trophozoite of Entamoeba. So for molecular analysis, successful DNA extraction and effective primer are important. Please remember, successful DNA extraction and we must select good primer. So how to extract the DNA? Of course, we collect the feces, and if possible, freeze. And for extraction DNA, we must do freeze thawing, because trophozoite is very easy to be disrupted, but cyst is very strong. That is why by freeze thawing, cyst can be disrupted. Then DNA can be released. That's why we had better do freeze thawing 3 or 5 times to disrupt the parasite, especially cyst. And then we collect supernatant after freeze thawing in PBS. We can extract the DNA by, here again, DNA struminicid or DNA zoo and others. We can select any preferable kit. And serial section. Maybe trophozoite or cyst can be contained. Then we can extract the DNA, for example, here again, DNA, FFP, tissue kit, and others. Then we can extract the DNA of the endomember from feces or serial section. So the other important point, how do we select good primer? Of course, we must study the information by published papers. Many papers we can see, but we must study for selection. As we explained previously, 3 species can be detected in pigs. For example, Endomember histolytica. This parasite infects human, as you know, which means that huge number of researchers pour much effort compared to those of animals, Endomember poreci or Swiss, of course. Thus, we can easily see many good and specific primer sets for detecting Endomember histolytica. That is why we can select the primer targeting 30 kilodalton cysteine-rich protein gene, and so on, some of them. That's why we can select some of them, including the primer here. So how about the other Endomember species? Yes, Endomember Swiss and poreci. I studied many published papers, so we could find 3 primer sets. One is E. poreci 1-2. This primer was reported by Belwege, Dr. Belwege, 2001. He described this primer was specific Endomember poreci, but we don't know all of the subtype could be detected or not. He just reported this is the specific primer for Endomember poreci. And Entom 1-2, also Dr. Belwege reported. He described this primer was genus, Endomember genus specific primer. Actually, Entom 1-2 were widely used in the world. Then, third primer, 764 RD3, 764 to 765. This is the nested primer. Dr. Clark reported. This primer were reported to be suitable for porcine Endomember. We don't know Endomember poreci or Swiss and so on. We just found these 3 primers. Then we had no information which primer were good or bad. That's right. So, but we can use one of 3 primer sets for analysis. First case is Nagasaki Prefecture. Nagasaki Prefecture is here of Kyushu Island. Osaka is here, Tokyo is here. This is Nagasaki. Nagasaki, this is Nagasaki map. Very complicated island, but Nagasaki is very beautiful of the harbor. I will talk about shortly about Nagasaki. In 1945, atomic bomb had been dropped here, following Hiroshima. Hiroshima is here, here. And the second atomic bomb had been dropped in Nagasaki. And Nagasaki Prefecture is very complicated form like this. But Nagasaki is nice prefecture. I have been there 4 times. So, situation of the pig farm at Nagasaki. In June 2013, more than 100 pigs showed refractory diarrhea, and some of them were dead. Veterinarian treated them by adding some medicine, but situation were not improved. We tried to do autopsy 3 dead piglets first for diagnosis. Autopsy of dead piglets, like this, edema, and we could see hemorrhage, and like this, or the bloodly contents like this. And number 2 of piglet, we can see also hemorrhage like this, Then veterinarian made histopathological sections, so they could successfully like this. You can see the disruption of epithelium cells, and these blood at red color are blood. That's why we can conclude hemorrhage, deciduous of epithelium tissue, and edema of submucosal tissue were observed. And then veterinarian could detect like this zoitis. If students could obtain this form, can you determine these are trophozoites of endomember or not? Maybe it's very difficult. Of course, in Japanese veterinarian cannot diagnosis, but luckily the veterinarian expect they were trophozoites. Then she asked me, what is this? Maybe a trophozoite, but we must confirm by molecular analysis or some staining. So then I stained by toluidine blue stain. We can see bloodly cell by staining. They are erythrocytes, and they are trophozoites of maybe endomember. And trophozoites engulf erythrocytes or bloodly cells. So electron microscopy, this is erythrocytes engulfed, and this is a nuclei. And interestingly, usually endomember doesn't possess mitochondria here, because in intestinal organ, O2 or oxygen population is very low, 5% and so on. That's why endomember lost mitochondria. But this parasite possesses mitochondria. Interesting. What is this? We don't know then. That's why, and additionally, veterinarian check other pathogenic agents. No main pathogenic bacteria were not were cultured. That's why the trophozoite could invade into lamina propria, which might cause hemorrhage colitis. First time, I thought this parasite, Entoamoeba histolytica, this can be first report for infection Entoamoeba histolytica in pig. I thought then. That's why I used the primer set EHL, EHR, this one, and P12, 11, 12, 30, 40. I used two primer sets for Entoamoeba histolytica. And then I selected this primer set because the nested primer is more sensitive, I thought then. That's as a result, for Entoamoeba histolytica were negative. But this primer set were positive here. So we could successfully see partial sequence were here. And the partial sequence were completely identical to those of Entoamoeba suis in gene bank. That's why we could successfully suggest Entoamoeba suis could invade into lamina propria in addition to Entoamoeba histolytica, then cause bloodily lethal diarrhea, like Entoamoeba histolytica in human. So, of course, Entoamoeba invaders in reptile can invade into lamina propria, but in reptile. So, only two species, Entoamoeba histolytica invaders, could invade into lamina propria in human or in reptile. But maybe Entoamoeba suis can invade into lamina propria in pigs. We could speculate they are so active because they can produce energy in mitochondria. So second cases, Shizuoka. Shizuoka is here, near to Tokyo. Shizuoka possesses Mount Fuji. Mount Fuji is so beautiful and very the highest mountain in Japan. So we can see Mount Fuji at Shizuoka, which is the highest and the most beautiful mountain in Japan. Here, so many Japanese green teas like this were produced at Shizuoka Prefecture. Very delicious. So, situation of pig farm. In September 2014, some piglets showed severe chronic and refractory diarrhea. Veterinarians have had difficulty to treat the piglets. We necropsied 3 animals. Here we can see swelling and horse-like sickling were observed in the affected ileum, and which turned red due to congestion, like this. The ileum contents are muddy or mushy stores, like this. Abnormal feces we could see. So the veterinarian of the local government at Shizuoka, they made a serious section of by pH staining. Of course, this is normal staining, but you can see no clear epithelium cells. That is why proliferative iritis was observed with severe inflammation of macrophage and neutrophils here. Then bilious atrophy and intestinal abnormal-like finding present here. And in the ulcerated areas, the intestinal epithelium was necrotic, and a large number of Entoamoeba species trophozoites were found in the lamina propria here. But maybe students could note, because the morphological phenotype were different from those of Entoamoeba suis, because these trophozoites could not engulf bloodly cell erythrocytes. And we can see some phagocytes, white color, like this. This one. Then trophozoite could engulf the bacteria here, like this, but not erythrocytes. Maybe I speculate they were not entamoeba Swiss, as previously reported. So interestingly, in bacteriological analysis, we can see many comma-like bacteria here by watching study staining. And I used anti-Lawsoria intestinalis monoclonal antibody. We can find many bacteria positive reaction of Lawsoria intestinalis. And then other main bacteria and virus were negative. That is why Lawsoria intestinalis is known to be an obligate intercellular gram-negative curve bacillus, which causes severe proliferative enteritis and characteristic intestinal adenoma in many animals, including pigs. That's why we could find Lawsoria intestinalis and entamoeba trophozoite, but we don't know which species of entamoeba. Then we extract the DNA from the section. So of course, I checked entamoeba histolytica by using 2 primers. And then I selected entamoeba polecki 1, 2, and the nested PCR. But about entamoeba histolytica of 2 primers, the result was negative. And in previous analysis, we could find entamoeba Swiss by this nested PCR. But in these cases, the nested PCR was negative. So I just use NA polecki 1, 2 reported by Dr. Bellucci. This is a positive. That's why I read the sequence of the PCR product. Then sequences were completely identical to those of ST1. That's why this parasite were found to be entamoeba polecki subtype 1. So I summarize shortly. In previous report, entamoeba Swiss and entamoeba polecki were less pathogenic or no pathogenic. But I reported entamoeba Swiss, this parasite possesses mitochondria to produce energy. That's why entamoeba Swiss can invade positively and actively. And then bloody diarrhea symptom. Otherwise, why Lawsoria intestinalis infect epithelium cells, then the barrier can be disrupted. So maybe entamoeba polecki, especially subtype 1, can be less pathogenic. But after disruption of the infection, Lawsoria intestinalis, then entamoeba invade easily into lamina propria because no barrier. That's why the disruption of epithelium cells as barrier, and the second, consequently, entamoeba polecki ST1 easily invade into lamina propria. Then it is difficult to cure by veterinarians. Antibiotics are not effective to entamoeba species, of course, and it is very difficult to treat affected animals. So finally, third cases, Aichi prefectures. Aichi possess many good castles like this, and there are so many famous castles in Aichi prefecture. Famous and very delicious broiler chicken. This is we call Nagoya Kochi. Very delicious and rare, which was called Nagoya Kochi like this. Yeah. So, situation of pig farm. In October 2014, some piglets showed severe chronic and refractory dark brown diarrhea. The same situation. and veterinarian treated them with enterofloxacin, but they are not improved. We necropsied 3 animals. So each prefecture ulcerative colitis with inflammatory cells in lamina propria like this. Surface are not clear. And submucosal tissue trophozoid in the deeper lamina propria at the ulcerative region were found. Then Lawsonia intestinalis were also detected. We could not find any mitochondria and engulfed erythrocytes. That's why I speculated this parasite could be Entamoeba polecki ST1. So we did molecular analysis. But unfortunately, we could not find any positive band. Entamoeba histolytica negative. Nested PCR for Entamoeba Swiss negative. And polecki 1-2, Entam 1-2 are also negative. What we will do? Then I checked all of the primer sets by alignment. So position of each used or published primer for porcine Entamoeba species. For example, Entam 1-2 reported by Belwegi 2003 here. By crack, nested PCR 764, 765 here. Very overlapped. E polecki 1-2 here like this. So maybe we can amplify by using these primers, but the result, all of the results were negative. So we checked not only position, the sequences. For example, this is Entam 1 sequences, forward primer and second primer. So subtype 3, subtype 1, 2, 4, Entamoeba Swiss. I compare all of 5 Entamoeba in pigs. So 3, 1, 2, 4 are almost identical. Dot means identical, the same. And dash means no data. Based on this comparison, Entam 1-2 very good. But Entam 1-2 could amplify the genes of other organs, especially bacteria. It can be known or less specific Entamoeba. Of course, Entamoeba can be amplified, but by many sequence analysis, we checked, but many bacteria could be detected. That's why this Entam 1-2 less specific. If we do not culture the Entamoeba species, isolate from the bacteria, we can do. But if we do not culture the Entamoeba, this Entam 1-2 are not to be suitable. In my opinion, this primer is not recommended. Then 764, 765, this is primer. This is primer. Then, interestingly, Entamoeba Swiss only 100% identical. But Entamoeba polecki 3, 1, 2, 4 different, completely different. That's why 764, 765 is suitable and specific to only Entamoeba Swiss. Okay. Finally, E. polecki 1-2. I compared. This is the primer. Entamoeba Swiss and 3, 1, 2, 4. You can see only subtype 1 are the same, and the other are different. That is why he reported E. polecki 1-2 was suitable for Entamoeba polecki. But based on these results, A-Polecki 1 to 2 is suitable and specific for only subtype 1. So in previous study, we are just lucky choose the good primer by chance. So how do we do? Yes, I try to design universal primer for Entomember Polecki, all subtypes. Then this is reported primer. Then I designed originally E. Polecki F1 R1 10 primer sets. So I checked all of the primers. So we could find the Entomember, E. Polecki F6, R6 were very good. For example, F6, R6 are identical, ST4213, like this. Maybe Entomember Swiss are similar, but maybe not amplified, only for ST124, all subtypes. Oh, sorry. So that is why this is summary protocols. We can extract the DNA by feces or section, and we can extract the DNA and PCR. If 764, 765 positive, we can detect Entomember Swiss successfully. But Entomember Polecki F6, R6 positive, we can do sequencing analysis. So we can differentiate successfully ST124 like this. That's why Entom 1-2, maybe ST1234, okay, but less specificity. This primer set can amplify the gene of bacteria. 764, 765 suitable for only Entomember Swiss. And then E. Polecki 1-2 is only ST1. So I designed E. Polecki F6, R6. We can amplify the genes from all of subtypes. Then exact diagnosis. So this protocol or strategies can be available for the other protozoa or parasite and bacteria viruses. So we must check all of the published primers, and we must compare the sequences like this. And then if no less specificity, we can design originality. But sometimes difficult to select a good one. If we are lucky, we could select one good primer. So finally, third topics. As you know, feces are so attractive world, like space. Of course, in the feces, so many bacteria or the DNA from the host and food and so on. That is why we have to design or select the specific primers from the pick up the DNA in the space or universe. So, for example, if I collected the feces from cattle or pig or chickens, of course, many parasites can be contained. If I or we extract the DNA from the tube and we conduct the PCR, and then we analyze the sequencing. So like this, many signals, we can determine the exact sequences. Of course, we can do TA cloning, the PCR product, insert the bacteria and cloning and sequencing. It's a little tough and complicated, but we can successfully read the clear sequence. But the sequence, only one sequence, which parasite we cannot determine. We cannot determine which parasite of the sequences. That's why I proposed this method. For example, many parasites, if we can see many parasites, of course, we purify oocysts by flotation method. And then, do you know this machine, embryo injection? So we pick up one oocyst by capillary, very small volume, and then pour into PCR tube, only one oocyst. Then freeze thawing, 99°, 5 minutes, and PCR and sequencing. If we can determine the sequence, we confirm this parasite, the sequence is for this parasite, and the sequence, this sequence is for this parasite, and so on. We can determine the exact sequence in each oocyst or cyst and parasite. Like this. This sequence is for this parasite. So this is a summary. I just introduced some examples of protozoan parasites in this lecture. You can utilize the same method and apply to other pathogens. The field as well as species are like the universe, and they can be parasites for us, a parasitologist. Paradise, they can be the paradise for us as a parasitologist. In near future, maybe we can easily analyze whole genome of the oocyst or parasite in each organism, maybe. But we can do, of course, we can obtain more interesting information. But so far, we can do PCR analysis or one-cell analysis and so on. But this analysis is important for exact treatment or exact purification, extraction, and selection of primers, good primers. Then all of the students can contribute to high production in livestock by many scientific methods. Okay, my lecture is finished. Terima kasih banyak, and thank you for your great attention.
Sorry, Professor Yunus, you're still in mute mode.
Okay, thank you, Professor Makoto, and nice presentation. And from that, and then there is the question, and now we enter to discussion session. I think so many questions to waiting what to the answer from you. And from then, if I look at the chatting box, I think some question from the participant. Okay, maybe you can answer directly after I read some the question. Yeah. First, the question from Francisca Okta from Sikia, Banyuwangi. Okay, so much, Professor. Regarding your share in the case 2, that of 60 days, I think, not 60 years old, 60 days old piglet. Can we include that Schistoisospora Swiss play role as the primary cause of the death? It means of Schistoisospora Swiss play role as the primary cause of the death. Is there any secondary infection by bacteria or virus? Maybe. Moreover, is Schistospora causing death on other species of host, such as cattle, poultry, dog, cat, or wild animal? Thank you very much, Professor. Maybe you can directly answer, Professor.
Okay, thank you very much for that nice question.
Yeah.
Yes, in these cases, in dead piglet, we checked main bacteria and viruses. That's why we can delete the possibility of bacteria and virus as a lethal pathogen. Then we maybe, of course, we didn't analyze all of the bacteria and virus. And we just check main pathogenic virus and bacteria. So it is possible that Cystoisospora could cause lethal symptoms. That's why maybe he or she asked me the main reason could be Cystoisospora or not.
Yes.
In our result, Cystospora suis could cause lethal symptom.
I see.
Yeah. And how about other questions?
Any other host? It's mean like cattle can dead by Cystospora.
Actually, Cystoisospora suis is specific for pigs only.
I see.
That's why, and Cystoisospora can infect only carnivora.
I see.
That's why cattle or chicken cannot be infected with Cystoisospora.
I see. I see. Okay, Sensei. I think it's everything clear about this question. And okay, the next question is also from Sikia, Audina Putri from Banyuwangi, some region of East Java province, from Sikia College. Regarding the entamoeba identification, can we differentiate cyst or trophozoid states between entamoeba coli and entamoeba invadens only with microscopic examination? Only microscopic examination. Because when we examine fecal sample from reptile, we frequently found cyst states of entamoeba. Is there any specific staining method in order to make cyst more contrast than the debris? Thank you.
Actually, we cannot differentiate the species of entamoeba, Coreki or Swiss and Histolytica. It's very difficult, but we can suspect based on the number of the nuclei in the system. The size is very similar. Oocyst size and as you know, trophozoid is not shape. That's why after cultivation or isolation, we can check the number of nuclei. But maybe it's very difficult. That's why we had better do PCR analysis for identification.
Maybe there is any the special staining to differentiate about it.
For example, iodine staining is good for the detection of entamoeba cyst. But this staining is not for differentiation of the species.
I see.
Just the cyst or
I think the condition like that because some iodine staining I think is to detect but not differentiate each other. Okay. The next question, I think we still have time, Dr. Aditya.
Yes, you can continue.
Okay. From Dr. Mufa, my colleague, Dr. Mufa. There is one question. Has it been attempted for the molecular identification of entamoeba in pigs by loop-mediated isothermal amplification, LAM? LAM. And then because this method is very possible to use in the areas with limited laboratory equipment. Thank you. Is there any possibility to detect the entamoeba by LAM? Yes.
We actually I just designed the primer set of PCR. So LAMP is very easy compared to those of PCR.
Yeah.
In the future or as a next step, we can try to construct the LAMP method.
I see.
Not yet, but not yet.
Yeah.
Good. Yes, and I agree that opinion.
Yeah. Thank you. Okay. Next question from your student, I think. From Dia. Dia,
okay.
You know?
Yeah.
Yeah, from Dia. Your information about the primer of apolacy, is it true that the primer apolacy F6 R6 more sensitive to S apolacy? In our current research, we found that some sample positive for primer F6 R6, but negative in primer apolacy 1 and 2. And the apolacy ST3, but when we tried to sequence the result apolacy 6 ST3 based on the gene bank. And this is the condition. And is any sensitivity between primal F1 R6 or F6 R6 and R6 F6 or R6. Yeah.
Dia asked the sensitivity of primer F6 and R6.
Yes. Yeah.
As you know, primer PCR is sensitive, but all of the gene cannot be detected by PCR because feces can reduce the amplification of PCR.
Yeah.
That is why we don't know exact sensitivity of F6 and R6.
Yeah.
Logically, F6 and R6 can amplify of all of the subtypes.
Yeah. Okay. I think the condition like that. Okay. The next question from Doni Krismanto. He is the Professor Nunu's son, I think. Okay. The result of my research found the presence of Endomember population in pig feces in the East Java, but I couldn't detect the presence of Endomember population in pig in Bali. How about that? The condition? Maybe there may be different location, different area. I think maybe. Okay.
It's very interesting result.
Yeah.
Actually, previously, I and Dr. April examined the Endomember in pig in Tangerang. So the prevalence was very high, and one of results by Dr. Nunu are also very high.
Yeah,
I think in Indonesia, Endomember Swiss could distribute widely. But if Dr. Nunuku find very low area, we had better focus on why. Maybe a cross contamination.
Yes.
Contamination.
Yes.
Good hygiene production.
Yeah. Maybe the condition of environment. I think maybe there is influence the environment condition in the infected by the maybe pig or cat or something like that. Okay, Sensei, and then I think the other question maybe I would like to ask some a little problem. What do you think about maybe the infection in the cattle, particularly in the influence of like Endomember, like the other Protozoan, Protozoan? Maybe you have experiences a lot of in the cattle Protozoan parasite. It's mean to be related with one cell analysis, something like
that. Actually, one cell analysis is for suitable for the molecular analysis.
Yes.
And if we can differentiate morphologically, this is torricollis or capillaria. In such a case, we don't have to do a molecular analysis.
I see.
That's why almost the same morphology.
I see.
Then, as the next step, we must do PCR or one-cell analysis.
I see. Yeah.
Morphology-
Maybe the problem is if we want to one cell analysis from the infection, the problem is to isolate the pure of pathogen, like if we take by squid, gentle squid and small squid, I think it's difficult to get it some the really this is a pure of infection. Sometimes there is the mixed infection, right?
Yes.
Like Amelia and cattle, so many species. Amelia, what do you think about it? If we find this case?
Do you ask how evaluate the pathogenicity of each isolate?
Yes.
Actually, it's very difficult, but we must do exact identify the species first. And then if I found some bloodly diarrhea and lethal diarrhea, and then we check the species. And then this is the pathogenic or not. That's why we must construct the data of the species.
I see. Yeah. Okay. Another question, I think, also from Dr. Doni also.
April.
Oh, Dr. April?
Dr. April.
Dr. April. Okay. This is from Sanjay, from your best friend, Sanjay. Dr. April. Okay. I'm sorry, Dr. April. We have previous experience with lamb for the detection of Trypanosoma M.Si. The probe is effective, although it is oversensitive and susceptible to contamination. In my opinion, we got a lot of false positive from the sample we tested at the lab. I agree that we should investigate the efficacy of lamb approach for Endomember analysis. That is like the Dr. April experiences is very sensitive to the other. Okay, please, Sanjay.
Yes, he suggests the lamp is better for the detection of parasites.
Yeah.
Yes, I think so.
Because oversensitive, Sanjay.
Yes. Then maybe I have not yet constructed the lamp method.
Yes.
Maybe in near future, Dr. April will try.
Yeah. I think I think the collaboration need to continue in the next time.
With Dr. Nunuk and so get together.
It's very important to
May
continue the collaboration.
May I have some comment, Prof?
Okay, please, Dr. April.
Yeah. I do agree with Dr. Mufassirin's suggestion for doing lamb for Endomember. But as I said, based on our experience at the laboratory, the method is oversensitive, to be honest. And we got easily contamination. Easily contamination. So that's why in some paper they said they wrote they published that lamb is better than PCR.
Combined.
That's correct, but we don't know whether they got the result because of false positive or false negative, something like
Yes.
that. Because when we tried in laboratory using some primers, we got confused. To be honest, we got confused.
Yeah.
Why we got the result like this?
Yeah.
That's why for in terms of Trypanosoma M.Si, I don't recommend to use LAM for detection, but we need to investigate the efficacy LAM for Endomember. Maybe the result better than in Trypanosoma M.Si.
Yeah.
I think it's a good idea if we collaborate to investigate this technique.
To become phenomenon, you know?
Yeah. Thank you.
I see. Okay, I think,
Prof, Prof Makoto. Sorry, Prof Yunus. Prof Makoto, many participants wants material of your slides today because your slide is interesting topic and nice presentation. Thank you.
I will send you.
Yes, by email. Yes, thank you very much.
Okay, how about the time, Dr. Adit?
Yes, you can finish your Q&A session. Yes, it's over.
Okay, thanks you, Professor Makoto. Your nice presentation and your answer is very good. If there is some many questions from the participant, please send your question personally by email to Professor Makoto Matsubayashi. I think from that guest lecture is very nice, and I think there's so many benefits for us, and then we want to continue this event in the other time, you know. And then, okay, please, from all participants, give a big applause to Professor Makoto Bayashi. Okay, from me as the moderator, as there is something mistake, please, I would like to apologize everything for me. And thank you, Dr. Adit. I return back to you. Okay.
Thank you very much, Professor Yunus, for guiding the guest lecture session. Now, before we move into the closing agenda, because there are still more than 170 participants. Wow, it's an amazing number. So please allow me once again to take a picture from all participants. So kindly remind, you can turn on your camera and I will start the countdown from 1 to 10. Okay, let's take a picture together. And 1, 2, 3, 4, 5, 6, 7, 8, 9, and last one, 10. Thank you very much. Also, special thanks to Professor Makoto Matsubayashi, all of committee and participants for attending today's guest lecture. Before I end this session, on behalf of Faculty of Veterinary Medicine Universitas Airlangga, I would like to invite Professor Dr. Nunuk Diartnolastuti, Doctor of Veterinary Medicine, Master of Science, to hand over the certificate for our remarkable speaker. To Professor Nunuk Diartnolastuti, the time is yours.
Thank you very much, Dr. Aditya. The Honorable Prof. Makoto Matsubayashi, thank you so much with your nice presentation and interesting of the topic today as a part of the Adjunct Professor activities today. On behalf of the Parasitologi Division and Faculty of Veterinary Medicine, we give a certificate as a thank you for Prof. Makoto. Please, the certificate, let's see on the monitor. Yes. Prof. Makoto for participant as speaker. Thank you very much. With the topic molecular identification of gastrointestinal protozoa parasite in livestock and successful trial toward one cell analysis. Yeah. Thank you so much, and we applause to Prof. Makoto. Thank you very much. Prof. Makoto, I'm speechless because you good lecture, yeah, nice lecture, and you gave a lecture of parasitology, especially of the protozoology. Maybe next time you gave about the helminthology or helminthology and the gastrointestinal tract. And because this topic interests for our student and lecturer, especially in the division of parasitology. And the topic is very useful for conducting research, especially of single cell protozoa is difficult to determination by microscopists. Yeah. And the method cannot distinguish for protozoa until species like Entamuba, Cystoisospora, Cryptosporidium, and so on, because they have very small size of protozoa. And thank you very much. You gave a lecture about molecular identification. And congratulations, Prof. Makoto. Your article have been published.
Yeah.
Yes, have been published in Parasitology International, and at your affiliation of UNAIR. Thank you so much as a part requirement of the Adjunct Professor. And thank you very much for all seminar participants, professor, lecturer from Faculty of Veterinary Medicine or other faculty, other institutions, especially for Dr. April from BRIN, and thank you very much for all students of undergraduate, master, and profession, and doctoral student for the meeting, yeah, for the attendance meeting in the parasitology lecture. Once again, thank you very much, and see you on the next seminar. Yeah. From Prof. Makoto, maybe we will invite you to give a lecture next time. Thank you very much. Arigato gozaimasu. Thank you.
Thank you. Arigato.
Yes.
Okay.
I think enough, Prof. Dr. Adit.
Yep. Thank you very much, Professor Nuno. And now the guest lecture session is finished. For Professor Makoto Matsubayashi, if you still have other activities, you can leave this Zoom meeting room. Once again, thank you for sharing your expertise with us today. Personally,
thank you.
I'd like
Bye
to say
bye,
thank
Prof.
you
Makoto.
to our speaker and participant.
Thank you, Prof. Makoto.
I do apologizing
Bapak Ibu,
to
terima
get any
kasih
mistake
semuanya.
or
Makasih,
technical problem.
Makasih, Dr. April.
Sukses
Terima
ya.
kasih. Amin. Salam sehat
Makasih,
semuanya.
Dr. April.
Oke.
Terima kasih.
Mohon lift.
Saya live,
Assalamualaikum.
Waalaikumsalam.
Thank you. Hopefully all of you will receive many scientific information and have nice day.
Yeah.
Good
Have a nice
afternoon.
day.
Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam. Bye-bye.
Bye-bye. Matur nuwun semuanya, terutama Prof Nunu, Dr. Adit.
Terima kasih sama-sama Bu Puji, Dr. Adit.
Sami-sami Dr. Puji. Ya, sehat selalu, sehat selalu.
Ya, sehat selalu ya.
Semangat ya, senang ya di Bogor ya. Terima kasih pada Prof Setiawan, Dr. Kusnoto, Dr. Agus, Dr. Endang, Prof Lucia ya tadi yang sudah memberikan.
Ya, dia juga terima kasih dia, kamu sudah hadir dia.
Banyak terima kasih. Semoga lain waktu kita bisa ketemu untuk acara webinar yang akan datang dalam kondisi sehat walafiat semuanya. Terima kasih.
Amin. Monggo.
Assalamualaikum.